I'm going to turn this portion of the webinar over to our moderator, Lauren Collins.
Good morning and good afternoon, everyone, and thank you for joining us for the final installment of the Methods series. Before we get started, I just want to reiterate that we'll be using the chat feature to communicate. So as you have questions arise during the presentation, please submit them through the chat feature. Make sure to be sending to host, panelists, and presenter. I will be keeping a parking lot of all the questions, and we will try to answer as many as we can in the order they came in. If for any reason we do not get to a question, those questions will be addressed offline. So please submit questions as they come to you. So without further ado, I'll turn it over to our presenter today, Dr. Hooper.
Hi, everybody. Thanks for joining me here today. This is a new experience for me. I don't believe I've ever done a webinar before, so we'll see how this goes. I like having at least being able to see myself to know if I'm doing anything embarrassing on screen. So the research that I'm going to be talking about today is related to some of my work with Eric Donnie and the CECTR project, where we developed some methodology to estimate causal effects for very low nicotine content cigarettes. As we'll see as we go through this presentation, is that there are some unique features of these data that prevented us from being able to apply a standard causal inference methodology to analyze these data, and I'll point those out as we go. Okay, so let's first sort of make sure everybody's on the same page with regards to the research question, and I'm going to go through this quickly because I know that a number of people here probably know more about this than I do. But we are going to be discussing if the evaluation of nicotine reduction is a potential regulatory strategy. So the FDA can implement a nicotine reduction policy. As part of this policy, the idea would be to reduce the nicotine content of cigarettes to levels that prevent the development and maintenance of dependence. But a key feature is that it can't be zero, right? Furthermore, we're pointing out that this is different than light cigarettes that increase ventilation, et cetera. So again, we're talking specifically about reducing the nicotine content of cigarettes. And in order to fully evaluate this as a potential regulatory strategy, we need to be able to understand the positive and negative consequences to sort of fully evaluate the public health impact of such a regulatory strategy. So that last part is going to be sort of the key point that we're trying to understand today, is sort of how do we evaluate this as a regulatory strategy and fully evaluate the public health impact. Okay, so let's think about the evidence related to nicotine reduction as a regulatory strategy, and I'll specifically be talking about the data that arise from Eric Donnie's CECTR project. So CECTR was a 6-week randomized trial of 839 daily smokers. The 839 smokers were randomized equally to 7 groups. There were 2 control conditions, a usual brand control condition, a normal nicotine control, and 5 experimental conditions with varying levels of reduced nicotine content. The lowest nicotine content was 0.4 milligrams per gram of tobacco as compared to 15.8 for our normal nicotine control. What we found with the trial is that the lowest nicotine content, the 0.4 milligrams per gram group, had significantly reduced nicotine exposure, nicotine dependence, and cigarette smoked in week 6 of the trial, right? So this is relative to the normal nicotine control. If you evaluated it compared to the usual brand control, you would have seen more or less similar results. So the key feature of this analysis, and what's going to sort of be the jumping off point for this discussion of estimation of causal effects, is to point out that this analysis was completed following the intention-to-treat principle, which is the sort of standard approach to analyzing data from randomized clinical trials. So what is an intention-to-treat analysis? So as I said, this is sort of the generally accepted approach to analyzing data from randomized clinical trials. So the idea of an intention-to-treat analysis is that subjects are analyzed according to their randomized treatment assignment, regardless of whether or not they complied to their treatment assignment. So if a subject was randomized to the lowest nicotine content group, but they did not comply. They smoked commercial cigarettes anyways. They would still be analyzed as part of the lowest nicotine group, sort of, again, regardless of that compliance status. So as I said, this is really the standard approach for analyzing therapeutic clinical trials. So when I say a therapeutic clinical trial, I am discussing a clinical trial where we might be evaluating a new treatment or a new drug. And that distinction is going to end up being important when we compare or think about intention to treat in a therapeutic clinical trial versus the context of tobacco regulatory science, which will be a slightly different. So one of the motivations for intention to treat is that the intention to treat analysis estimates what's known as the use effectiveness of a new treatment. So the use effectiveness of a new treatment is going to be the effect of a treatment when used in an environment similar to the clinical trial environment. So the idea here being that if you are evaluating a new drug or a new treatment, in the current environment, some subjects will not comply to their treatment, and the same would be true when you actually went to use the treatment in practice. So therefore, the effect you observed in the clinical trial should be similar to the average effect observed in the normal population, where people may or may not comply to the treatment that they are prescribed. So I have some conceptual diagrams that I'd like to go through, which I think sort of will highlight why intention to treat makes sense in the context of a therapeutic clinical trial, and then perhaps use that to motivate why it may not make sense in the context of tobacco regulatory science, and specifically randomized trials of very low nicotine content cigarettes. So this conceptual diagram is for the therapeutic setting, so the intention to treat analysis in the context of a therapeutic randomized clinical trial. So on the left is the basically your current population. So this is the population that is used, that we're sort of currently in, and that we will be running the clinical trial, right? So the key point here is that in the context of, let's say, a new treatment that might be under an IND, so like a new drug, or a device that hasn't been approved yet, typically we are in a setting where the subjects don't have access to that treatment, right? So if it's a new drug and it hasn't been approved yet, in general, people don't have access to the treatment. So our clinical trial, which is this next component here, is going to be a sample from the population. Now, I didn't necessarily say random, because the subjects that are enrolled in a clinical trial may not necessarily be random. Typically there's going to be some selection bias about who exactly enrolled in the clinical trial. But then within the clinical trial, they're going to be randomized to a treatment group, and I've simplified this diagram to basically just deal with one treatment group here for a second. So this, let's say, subjects have been randomized to the treatment group in this clinical trial. So in the clinical trial, the subjects that receive the treatment they're randomized to treatment. You add treatment. Say they're given the treatment. Some of the subjects will comply to the treatment they receive. Others will not. And then we use these data to estimate basically our intention to treat estimate. Here I've used theta to just sort of represent whatever it is we might want to estimate, whether it be a hazard ratio or an odds ratio or a difference in the means. It could be anything. I'm writing this specifically in general. So again, we've taken these data and we sort of take the compliers and the non-compliers. We basically analyze everything regardless of whether or not they complied, and we get our intention to treat analysis. Okay, so then we move to our future population. So this might be our future population, and this is the population we want to be able to draw inference about, right? We really would like to know what would happen in this future population when people are actually able to be prescribed this treatment. So you take your future population, you basically add the treatment into the population. People are going to be prescribed the new treatment. Some will comply to their, what just happened there? Does everybody else see that? I've lost my figure. Okay, so not everybody will comply to the treatment, and so some people will comply, others will not comply. So therefore, the effect of the new treatment in this future population, we're just going to call that theta future. And in the context of a therapeutic clinical trial, the ITT estimate will converge to theta's future. So what I mean by that is basically, if you give me an infinitely large clinical trial, I'll get the right answer. So basically, theta ITT is going to estimate the effects of your treatment in this future population where people may or may not comply. So let's contrast that quickly with the case of an ITT to evaluate very low nicotine content cigarettes, so specifically to evaluate a nicotine reduction policy. So again, the idea here is that the FDA could potentially reduce the nicotine contents of cigarettes to some level greater than 0, but low enough to decrease or to prevent dependence from being developed. So this is our current population. Right, and you'll see here, in the current population, of course, people have access to normal nicotine content cigarettes. So that is, they have access to commercially available cigarettes with normal nicotine content. So we're going to run our clinical trial in this sort of context. You're going to then randomize some people to receive very low nicotine content cigarettes. Some will comply, others will not. So in this case, the non-compliers are going to be people that smoke commercially available cigarettes. The compliers are going to be people that only smoke the very low nicotine content cigarettes. So this gives us our ITT estimate. Now let's think about a future population where this kind of regulation might actually be implemented. Now, in this case, not only would you be sort of giving people these very low nicotine content cigarettes, but you'd also be removing access to the normal nicotine content cigarettes. Now, I should point out here, of course, this is a bit of a simplification. We're basically pretending here that there's no black market. Now, in reality, there would likely be a black market, but for the purpose of this conceptual diagram and motivating the discussion, we're not going to deal with that, and I'll sort of talk about that as a limitation at the end of the talk. So again here, the key is, unlike the therapeutic clinical trial case where all you're doing is sort of adding this treatment, in the context of nicotine reduction as a regulatory policy, not only are you sort of basically, you're not adding, you're really replacing. You're taking away access to the normal nicotine content cigarettes. So as a result, now we're thinking everybody will comply, basically because they'd be complying by regulation, sort of mandated compliance. That would give us some theta future. So the problem here is that now we don't really know the relationship between the intention to treat estimate and the future effect, right? This doesn't mean that they could be the same. There's not saying that they're not. But the point is the relationship is less clear, whereas in this case, it's clear that we will get a consistent estimate. So a consistent estimate basically means that we'll get the right answer with infinite amounts of data. In this context, it's not clear that that will happen. So the ITT might be estimating something different than what would happen in a future world where compliance is mandated by regulation. So the thing, or sort of the obvious thing that people might want to do would be a so-called per-protocol analysis. So what that means, so in a per-protocol analysis, what I've done here is, the future population hasn't changed, but what I've done is maybe I just look at the compliers, right? So I grade out the non-compliers. So I'm only going to use the compliers, and I get my per-protocol estimate. So per-protocol basically means I'm only using the people that followed the protocol, and I can use that as my estimate of the treatment effect. Well, the problem with that here is, and I've written this, I realize in some mathematical notation, but I'll say what I mean here. In this case, I've now represented X could be some set of covariates that might be a confounder. So that is, this might be things like age, sex, race, dependence, any number of measures that might be confounders, right? That might be associated with sort of this treatment effect. And if the issue is that so this right here is the distribution of the confounders for the compliant subjects, and this is the distribution of X in the overall population. Now, if they're the same, the per-protocol estimate will actually work. So that is, if these people look the same as the future population, then we're fine. But the problem is this is very unlikely. It is likely the case that subjects that complied with the treatment look different than the general population. And if that's the case, we can't use per protocol. The per protocol will give us basically some kind of incorrect estimate of a future treatment effect where compliance is mandated by regulation. So that's sort of going to motivate the use of more complex causal machinery, is this problem here where we basically have confounding. So that is, X is going to be something that's related to both compliance, but also to the outcome you're interested in evaluating, whether it be cigarette smoke per day, measures of dependence, measures of withdrawal, etc. So that's sort of, I hope this conceptual diagram sort of illustrated the motivation for what we're doing here. So the key thing we want to do is we want to try to get an estimate of this theta future, so this effect of VLNC cigarettes for this future population where compliance is mandated. So that specifies it here. So as I said, just sort of to highlight what we're talking about, our target of inference is the effect of nicotine reduction in a future regulatory environment where nicotine reduction is mandated. So in some ways, this is something we already know how to do. There are lots of statistical methods that exist for estimating causal effects in the presence of noncompliance, whether it be inverse probability weighting, principal stratification, structural nested equation models, instrumental variables. You might have heard of some of these things, or in fact be familiar with them, but these are sort of existing methodologies that we know generally work for estimating causal effects in the presence of noncompliance. So what's the problem? Well, in general, if we think about the methods that I just listed, so inverse probability weighting, principal stratification, etc., in general, these methods all assume that compliance is measured without error. So that is that you have a good measure of whether or not subjects were compliant. Excuse me, getting over a cold. So in the case of CENIC, in the case of this randomized trial of very low nicotine content cigarettes, that wasn't really the case. So there was a follow-up paper by Natalie Nardone who looked at the basically compliance in the study, and we found that amongst the subjects that were randomized to the lowest nicotine content cigarettes, 39% self-reported noncompliance, but when we looked at biomarkers of nicotine exposures, 75% of the subjects that self-reported, or 75% of the 60% of subjects that reported compliance had biomarkers of nicotine exposures, specifically TNEs, total nicotine equivalent, above a pre-specified threshold that has been proposed as a marker for full compliance, right? So the point here is self-report is really not a reliable measure of noncompliance. Sort of a bit wordy there, but the key point is we were able to verify that the majority of the people that said they were compliant actually weren't compliant. So the alternate approach we want to use, of course, is to use biomarkers of nicotine exposure with TNEs, cotinine, et cetera, and use these to identify compliers. Now, you could, in theory, take these and sort of use some kind of strict cutoff and sort of come up with a binary classification of compliance based on biomarkers of nicotine exposure, and then apply the existing methodologies that I highlighted earlier. But the problem with that is there is a potential for misclassification. That is that some of the people that you classify as compliant perhaps were not compliant, and vice versa. So we'd like to have a methodology that allows us to estimate the causal effects while being able to account for this potential misclassification due to using an imprecise measure of compliance. As I said, our objective is going to be to develop a statistical approach for estimating causal effects with an error-prone measure of compliance. And the way we're going to do that is we're going to adapt inverse probability weighting, so IPW, to accommodate imprecise measures of compliance. And you'll see how we do this in just a minute. So first of all, I do need to go through a little bit of notation here, just to sort of make sure everybody's on the same page here. And the way we're going to set this up, at least in the causal inference world, is to think about potential outcomes. So this is the basic sort of counterfactual framework where we're going to evaluate the potential outcome dependent on whether or not a subject was compliant. So first of all, we're going to define Z to be sort of the true number of non-study cigarettes that were smoked. So again, this is the truth, which we will often not be able to actually observe. So Z equals 0 indicates full compliance. So if the subject did not smoke any non-study cigarettes, then they were compliant to their randomized treatment assignment. C is a binary indicator of compliance. So that is, Z is a variable of zeros and ones that indicates whether or not a subject was compliant. And again, this is not directly observed. So we don't necessarily get to observe these data because some subjects will tell us that they were compliant when they were actually not. So in that sense, this is not fully observed. So Y star Z is going to be the number of study cigarettes smoked per day. So again, I should back up a little bit here and say we're talking about in week 6 of the trial. So this is the number of study cigarettes smoked per day as measured by IVR was what we used. So this is the number of study cigarettes smoked per day if we set Z equal to little z. So this is a potential outcome, right? Because in practice, we only get to observe Y star Z for one value of Z, right? But we can think about Y star Z as a sort of potential outcome for all potential values of Z that could have been observed. So basically the idea would be here that Y star 0 would be the number of study cigarettes smoked per day if, possibly contrary to fact, the subject smoked 0 non-study cigarettes per day. So the whole point of going through this is that our goal here is to estimate mu, which is the expected value of Y star 0, that is the average number of cigarettes smoked per day if all subjects were compliant. So that is if we sort of force them to be compliant. So that sort of is this potential outcomes framework. As I said, there is some counterfactual situations going on here as well. So thinking a little bit about the observed data, so it's important to point out that this is a little bit different potentially than the potential outcomes. So here, Y is the number of study cigarettes smoked per day. D is self-reported compliance, so this is going to be subjects, that is if they reported compliance, so that's a binary indicator. X are going to be confounders, and we're writing this in generality right now, but this could be things like age, race, dependence, so it's a lot of measures of sort of things like dependence and craving and baseline. Any of these things could be potential confounders. And B is going to be our biomarker, so i.e. total nicotine equivalent. So this is the standard inverse probability weighted estimator, right? So the idea here is as I've written it in somewhat formal notation, it's mu hat is our, in this case would be our IPW estimator, which is the solution to this equation. But the key point here is to consider this weighting. So there's this inverse probability of weighting thing going on, right? So C sub I is an indicator you were compliant. So again, we're only including the subjects that were actually compliant. And then you weight this based on the probability that you were compliant given your set of covariates. So that is if you have a low probability of compliance, so that is if subjects with your similar covariates have a low probability of compliance, you are actually getting upweighted in the data. If you are someone that's based on their conditional on their covariates had a high probability of compliance, you would be downweighted in the data. So the idea basically here is to effectively create something, a pseudo-population that kind of looks like the population you're trying to draw an inference about. So somebody just asked about exchangeability. I'll talk about those assumptions in just a minute. But yes, we'll exactly get to that. So that would be our standard inverse probability weighted estimator. So how do we rig this up to deal with something where C is not observed? So the key thing that's interesting about this is that C sub I is going to not be observed. So how would we do this when C sub I is not directly observed? So we've come up with this. We get the award for the most tortured acronym of the day. My colleague came up with this. It's very important in statistics to have a good acronym, so we're calling it the CURE estimator. So as you'll see, the only thing that we've really done here is that we've replaced C sub I with the expectation of C sub I, so the probability you were compliant conditional on the biomarker, X sub I, and then Y sub I. So Y sub I is our outcome. So basically, if you take C sub I, which is this unobserved probability, and replace it with this probability of compliance conditional on the biomarker, the covariates, and the Y, we come up with this new estimator that basically will allow us to get an estimate of the causal effect without actually observing C. Now, as you'll see, this introduces some new things that we need to figure out how to estimate, and I will talk about those shortly. So this is going to be our new estimator. So the key thing here is if we can figure out the probability that you were actually compliant, then we can replace C sub I with that probability you were compliant and get a consistent estimate. So the theoretical properties, and this is getting back to that question about assumptions. So basically, here we're thinking about a couple of different things. So the CURE estimator is going to be consistent. So that is, it will converge to the correct answer with an infinite sample size under the following assumptions. So A, we're going to assume no unmeasured confounding, right? So that is that basically X captures all possible confounders, and at the end of the talk I'll sort of highlight this as a potential limitation, is that this is a fairly strong assumption so that we can't have any unmeasured confounding. So positivity, so we're assuming that the probability that any subject was compliant conditional on their covariates was greater than 0. So that is, we don't have some subpopulation that had no probability of being compliant, or that a probability of compliance equal to 0. And then sort of consistency, so that is, we're assuming that if Y equals Y star 0, so Y equals Y star 0, if Z is 0. So that is, for the subjects that were compliant, what we observed would be equal to what the potential outcome when we set Z equal to 0. So these are sort of our three assumptions that we have to make in order for this estimator to work. Okay. So if you were paying attention here, you probably, one thing you might think here is, okay, that's great. We've proposed this new estimator here. We've proposed replacing C sub I with this new function that's a probability of compliance conditional on the biomarkers, the covariates, and the outcome. But the big problem, of course, is how do we get these functions, right? I mean, how do we get this? We don't, it's not like this is known. We don't know what the expectation of C sub I is conditional on B, X, and Y. Furthermore, it seems like this would be a very hard thing to estimate if you don't get C. So how do we do this? Now, of course, I'd be a bit of a snake oil salesman if I stopped the talk right now and said we can't. So clearly we do have an approach, and we'll sort of walk through how you might think about doing that. And it turns out that having a biomarker that discriminates between compliers and non-compliers will be quite important in order for this to work in small sample sizes. So let's think about a biomarker, right? So let's say we had a biomarker that we think will discriminate between compliers and non-compliers, like total nicotine equivalence, right? So if we can think about the distribution of B conditional on the covariates and the outcome, right? So that would be basically what is the, if you tell me a person's, age, sex, race, dependence at baseline, all sorts of other things related to their smoking characteristics, and then their outcome, right? We ought to be able to figure out what we might have some distribution for B, right? So the way we're going to do this is that we can decompose this distribution of the biomarker into a subdensity for compliers and a subdensity for non-compliers. So that is, if you had the distribution of total nicotine equivalence for all subjects at the end of the study, you would have lower values, presumably, for the people that were compliant and higher values for the people that were non-compliant. So this is known as a mixture distribution. So really, we can decompose this distribution into compliers and non-compliers. In general, this can be hard, but we do have some additional data that we can use to sort of augment our estimator with data from known compliers. So that's going to be important, and I'll talk a little bit about how that actually works. So again, the key point is we're going to write this as a mixture distribution, and when we do that, what ends up falling out at the end is that we can directly estimate this probability of compliance. So as I said, we write this as a function of these subdensities, and the implication is that this expectation of C given B, X, and Y can be estimated using only observed data, which is really cool because we're able to do this without having C. So again, C is still unobserved, so we are able to estimate this fairly complex function without actually observing C. So how does that work? So here is that mixture distribution I was talking about a second ago, and I'll just sort of highlight the different components so you can understand what I'm talking about. So this is the distribution of the biomarker. Right? So if we think about this as like a histogram generalized to a more smooth curve. So this is the density of the biomarker. So this is the biomarker for the compliers, or the density for the compliers. This is the density of the non-compliers. And here is the probability that a subject was compliant. So everything here is conditional on X and Y. So the key thing here is that if you actually had compliance, this would be easy, right? You'd be able to estimate this, and you'd be able to estimate this, and it would be no problem. But we do know in general how to estimate these things even without knowing C. There's something called the EM algorithm that can be implemented that will allow us to estimate this mixture distribution without actually observing C. So the primary reason this works is this somewhat neat trick where we can rewrite this probability, or this sort of probability that we're compliant, conditional on B, X, and Y as the components of this mixture distribution, which is sort of what I've done here. Now, this is somewhat, I don't mean to go into too much of the weeds here, but I was very excited that we were able to come to this realization because I found this to be a fairly elegant solution to the problem. So that is, we're taking something that would appear to be very hard to estimate, and by rewriting it using Bayes' rule, we're now able to write this as a function of things we can estimate. So the key point is we can estimate these four components. That's not a big deal. We know how to do that. And therefore, once I get those components, you can tell me, I can tell you the probability that a subject was compliant conditional on their biomarkers X and Y. So that is that if you give me a subject's biomarker at the end of the study, Y, so this would be their self-reported study cigarette smoked per day, and then the covariates, I can tell you the probability that subject was compliant. And this is mostly going to depend on B and Y. So if you think about it for a second here, if you've told me how many study cigarettes you smoke per day, we ought to have a pretty good idea of what your biomarker should look like. So by sort of comparing what your biomarker actually is to sort of one of these densities, we're able to basically determine how likely it is you are actually compliant. So if your biomarker is low, you're more likely to be compliant. If it's high, you're less likely to be compliant. So the key part about this is this allows us to estimate this fairly complex thing using only observed data. So a couple points on this. So in general, estimating mixture distributions is hard, even with the EM algorithm. It can be a challenging thing to do to sort of be able to identify these subdensities. So the way that we were able to do this was by augmenting our estimator with data from known compliers. So these are the data that were reported in Denlinger et al. So this was 23 smokers that were sort of sequestered in a hotel room for a week that were only allowed access to cigarettes with 0.4 milligrams per gram of tobacco. And then the biomarkers were measured on these subjects. So that when these data were published, this allowed us to basically estimate, get some idea of what the distribution of the biomarkers should look like in subjects that were fully compliant. So this is a completely separate thing, right? But the key point is that these data provide us important information on sort of what the distribution in compliers look like. So that means when we're trying to decompose the distribution for the entire population, we already know what one of the components should look like. which makes it a lot easier to sort of pull out the two components. So that was sort of an important point. Okay, so let me just show you the result of all this stuff. So I've gone through a lot of math, so let me just show you the result of this. So what's on the left here? This is a histogram of the log of total nicotine equivalents from the lowest nicotine groups, actually the two lowest nicotine groups from Scenic Project 1. So we had the 0.4 milligram per gram of tobacco group. We also had a group that had 0.4 milligrams per gram of tobacco with high tar, and we collapsed them together in order to have more data. So this is the distribution of the log of total nicotine equivalents in these subjects. And you can kind of see the two peaks of the distribution, right? So you have a lower peak down here. So this is on the log scale, so that's why you're getting the somewhat funky values. But you have a peak down here. Sure. I'll get to that once I sort of talk about fitting this in a second here. So this is our histogram. So we have, this would be our peak for the subjects with the lower values. So this would be the peak for the compliers. And then we have this peak for a higher value for the non-compliers. Now, in this case, getting to the question about the parametric versus non-parametric, so these would be log transform data. We considered a couple of different things. We considered a log normal distribution as well as a gamma distribution on the original scale. So we were doing things that were parametric. Non-parametric would simply not work with these data. We wouldn't have enough data. So we basically fit a log normal distribution, and we did do some checking to determine whether or not the data fit, the model assumptions were met, and we did not find any violations of our parametric assumptions. So we went ahead with sort of this log normal distribution, so a normal distribution on the log scale. Right. So this sort of illustrates it nicely. So the idea here is that if you have this peak here of the compliers, you have the peak for the non-compliers. If you look closely, there is this sort of dotted curve. This would be the subdensity for the compliers. This dashed curve, this dashed curve here would be the density for the non-compliers. And then this yellow line is this resulting mixture density that I was talking about, right? So we have this mixture density, and we were able to decompose it into these two sub-densities that you can sort of see in this figure. So the idea here is if we go back to what we wanted to estimate, the dotted line is the distribution of the compliers. The dashed line is the distribution in the non-compliers. So then using Bayes' rule, we are able to convert this into a probability that you were compliant. So that is, if you give me your biomarker value and the number of self-reported study cigarettes per day, I can give you the probability that that subject was compliant. So in the Denlinger et al. Paper, they proposed a threshold of 6.41. So at that point, 6.41 was the 95th percentile of the distribution in the known compliers. So if you were to look at that same thing here when we've done this sort of more complex modeling, so if this is about 6.4, we see that that actually ends up being about a 50-50 mark for compliance. So that doesn't conflict with the Denlinger et al. Paper. That was simply looking at basically the probability you being above that threshold if you were compliant. We're trying to flip this around and saying what's the probability you were compliant if you were actually at that value or lower. So this allows us to map your biomarker values to a probability of compliance. So again, if you had very low biomarker values, it's unlikely that you were compliant, or if you have low biomarker values, you were very likely to be compliant. High biomarker values meant you were unlikely to be compliant. And the key thing here is if you thought 6.41, if this is the threshold you were using, there's still a relatively reasonable probability that you could be above that threshold and still be compliant. Here is up around 8. We're seeing there's still a 20% probability that a subject was actually compliant. So this is exactly the kind of misclassification that I was talking about. Okay, so that was for the numerator, which becomes the hard part to estimate in our CURE estimator. The denominator is just a matter of fitting a regression model. So we did a general, basically a GLM with a logit link, using this estimated value, expected value of C given B, X, and Y. We use that as our response, and we just basically do a regression model. And this can be implemented using standard statistical software. So this is actually analogous to what you would typically do in inverse probability weighting. In inverse probability of weighting, you do a regression model for C. Now we're doing a regression model for the probability of compliance. So something a little bit different here. Okay, so now we've gone through all of this. So now let's look at the outcomes of this. So basically, what does this actually mean for the data from scenic? So what I've done here is I've taken a couple of different outcomes, and I've estimated the treatment effect. So this is the effect of the lowest nicotine content group versus the 15.8 groups that are our normal nicotine content controls. And I've looked at a couple of different outcomes based on a couple different ways to analyze the data. So there's the intention to treat. So this would be analogous to what was reported in the primary paper, albeit a little bit different because we are comparing the two lowest nicotine groups combined. The 15.8 group in the primary paper, we treated them as separate, but we're doing the combination thing because when we look at compliers, it ends up being very different. There's not enough compliers if you just look at one. So this is the intention to treat analysis. This would be a per protocol analysis if you just use the T&E cutoff from Denlinger et al. So if you use 6.41 as a hard cutoff for compliance, and you said anybody below that was compliant, anybody above was not, and then with our estimators. So looking at cigarettes per day, we see a relatively similar outcome for these different measures, although you do see that the per protocol effect is a little bigger than the cure estimator. So this is the ITT, it's about minus 5.89. If you do the per protocol, it gets bigger, negative 7.43. But with our cure estimator, it's a little bit attenuated back towards the ITT. I don't have a good explanation. We're still sort of digging through these data to get an idea of what might be a good explanation for these mechanisms. But this would be sort of what we saw for cigarettes per day. So if you look at the 95% confidence intervals, so these are confidence intervals from the non-parametric bootstrap, we can still exclude zero. So in some way, we still have a significant effect for cigarettes per day, even as you force compliance. So then I looked at FTND and MNWS, so measures of dependence and withdrawal. We didn't see a whole lot here as well, so this is actually reassuring. And if anything, the effect on dependence was a little bit bigger than the ITT effect, although we do see what the trade-off is here, right? So notice here that for FTND, for the ITT, we're able to exclude zero, i.e. have a significant result, but we're not able to have that for the cure. So this sort of illustrates that there's no free lunch, right? So we've proposed a lot of complex machinery, and as a result, the cure estimator will be less efficient and lead to wider confidence intervals than the ITT. Although it should be noted that in general, it's not going to be the case in this case, but in general, we found it actually to be more efficient than this per protocol approach. MNWS, similar things. We get fairly consistent results, although we actually see that withdrawal symptoms would have been, had you just done the per protocol, you would have concluded that it was higher in the low nicotine groups. But once we do the cure estimator, we get more or less the same as the ITT. But weight gain is one that I wanted to highlight. So this is one that's been explored a little bit by a paper by Laura Ruprecht. But this is looking at what would happen if we actually do this causal estimator, right? So this is the weight gain via the intention to treat. We see that there's no effect of weight gain. So subjects in the treatment groups had the same weight gain as subjects in the control groups. Now, if we do per protocol, we actually see that the effect is about 1 kilogram. So that is subjects in the treatment group gained an additional kilogram relative to the control groups after only 6 weeks. But in this case, we can't exclude zero. But then when we look at our cure estimator, the treatment effect is even bigger than the per protocol. It's now 1.4 kilograms, and we can actually exclude zero. So this sort of highlights the value and importance of looking at this for some of these unintended consequences. So the unintended consequence here being masked by the ITT analysis, but then when we look at the causal effect, we see that there is something that's certainly deserving of further investigation. So that's just sort of illustrating the results. So another point here, so that's sort of this was sort of our application. I also wanted to give you a little bit of data on does this work. I'm sort of running a little short on time, so I won't go through this too much. But as I said earlier, the CECTR estimator works in infinite sample sizes. That tends to be what we first look at in statistics, so that is as n goes to infinity. But real life, we don't have infinite data, so what happens in the sample size is similar to what we actually had in the scenic trial. And the way we do that is we're going to simulate some data under some different scenarios, and we're going to evaluate the bias and the mean squared error. So this is a rather imposing table of data. So I'm just going to focus on up here and sort of state that so this is the performance for a sample size of 225 per group. So this is similar to what we have if we combine the two lowest nicotine groups in scenic, and this would be larger sample sizes. And the key results to look at are this. So this is the bias, and what we have here are a couple of different estimators, so a per protocol and a self-report IPW. So we sort of know we wouldn't do these, but this is just to illustrate why they don't work. So these would have substantial bias. We want our bias to be 0. I'll come back to this IPW a little bit later. But then if you look at our CECTR estimator, the bias is much smaller, but it is a function of the quality of the biomarker. So what we have here is the area under the curve for the biomarker. So basically, the better discriminator of the biomarker, the better CECTR works. Now, TNEs actually has an area under the curve probably much higher than this, but this is important to know because AUC of 0.9, I have another student that's sort of looking at the area under the curve for some of these biomarkers. This might be something similar to what we see for anatabi, which will become more important when we start to look at these in conjunction with e-cigarettes. So that's another biomarker we can use to evaluate compliance. So it was important to see that we're still getting good results with the types of biomarkers, or basically with the discriminative ability of the biomarkers that we might have in future trials. Another thing to look at is this mean squared error. All I want to point out here is this cutoff IPW is equivalent to just using the cutoff from Denlinger et al., and our approach is more efficient. So it turns out that if you just use the approach done, say, IPW with the cutoff from Denlinger et al., you would have been unbiased, but our approach will be more efficient and not to a non-trivial extent. I mean, we're seeing a 25% reduction in the MSC. Now back to this IPW, the reason this is here is to illustrate what would have happened, how much are we losing versus actually observing compliance, and the reality is we're losing quite a bit. So that's just sort of a reality check to understand what would happen if we actually did know the truth about compliance rather than trying to have it from a biomarker. And the case is that we would certainly be better off if we didn't have to use the biomarker, although these results are still acceptable, and these results are impossible. These results are more just allowing us to frame our results, giving us sort of a best-case scenario. So the take-home message is that the performance was good, and certainly as n gets larger, the performance gets better. Okay, so finally, some concluding remarks. We've developed a framework for estimating causal effects with an imprecise measure of compliance. It appears to work both in finite sample sizes using our simulation studies or infinite sample sizes based on our theoretical results. We were able to estimate the causal effect of nicotine reduction when subjects are compelled to comply. The results remain promising, but unintended consequences should be investigated, so I think this will be a very important tool for evaluating unintended consequences. As I said, and as a result, it will be an important tool for understanding the impact of regulatory action. Some limitations. So anytime you use the causal machinery, there's going to be sort of no unmeasured confounding. Anytime we use the causal machinery, we have to make strong assumptions. I think the one here I would be most concerned about would be no unmeasured confounding, which I think is always going to be the case with these. I did mention early that in reality a black market would likely exist. So the question here is, I think you'd have to think about how you would use these results to model future. We are not really able to estimate the effect with, a little bit of compliance. So I think the way you would use this you'd give these results in conjunction with the ITT results to the population modelers and allow them to sort of understand, to sort of simulate what would happen if you saw, deviations in these effects due to a little bit of a black market. We in this case, only considered compliance in week 6 of the study. So in some sense, we were allowing people to be non-compliant for 5 weeks as long as they were compliant in week 6, which is a little bit weird. But that's something that we're sort of working on for the future. Mixture distributions can be hard to fit without the auxiliary data. This is something that we're currently looking into a little bit, especially as it relates to the intermediate dose levels, and I will comment on that in a second. So a couple things, future work. We actually have proposed or are currently developing some estimators that we think will be more efficient than the CURE estimator, so that will be important as we try to squeeze basically as much as we can out of these valuable data that are being collected in these trials. The trade-off, of course, is that when we have, in order to gain the efficiency, we're going to probably have to make even stronger assumptions. So we'll have to evaluate the robustness of these alternate estimators, as well as the CURE estimator. So finally, a lot of my research also sort of focuses around Bayesian approaches. I'm particularly interested in sort of borrowing strength across trials as well as borrowing strength across dose levels. So I am working in conjunction with a student on a method that I think will allow us to estimate causal effects for some of these intermediate dose levels where we don't have the auxiliary data. That's something we're currently working on, as well as borrowing strength across trials. There are multiple trials being run right now evaluating sort of similar strategies. We'd like to be able to combine them in a sensible manner. All right, quickly some acknowledgments. This is joint work with David Vach and Jeff Boatman. David is my collaborator at the University of Minnesota. Jeff is our student. Eric Donnie at Pittsburgh, who's the PI of Scenic, and then as well as sort of some information about funding. Thank you. So I guess we have a few minutes for questions. Alicia, is that how this is going to work?
Yes, it is. We're going to have our moderator, Lauren Katz, review those with us now.
So there were several questions that came through. Some of them have already been addressed during the presentation, but a few have not. First off, It seems this technique can be used only if you have a clinical trial. Is this correct?
Well, no, not necessarily. I think the question then you'd have to just, no. So, I mean, as long as you can make a reasonable assumption about this no unmeasured confounders, you'd be able to use it. But we'd have to think a little bit about what that would actually look like. So in an observational study, I think you just have to be more concerned about the covariates. We haven't thought about that yet for this application, so we'd have to think a little bit about what changes we might have to make if we're implementing this in the context of an observational study. But the framework itself, I think, has potential to work in observational studies as well. But as with anything, some additional work is probably needed to gain the best results in that application.
If there are any more questions, please feel free to chat them in. We still have a couple minutes. While we wait for any additional questions, I would like to make an announcement for our next webinar session, which will be on March 15th with Dr. David Ashley. He will be presenting the same presentation that he had given at the in-person T-Course meeting. So for those who are not able to attend, this is a great opportunity, and for those who were there, it's a great refresher. His presentation is entitled How Scientific Research Informs FDA Regulatory Action. This will be again on Wednesday, March 15th, and we will be back to our usual time of 12 p.m. Eastern. So please look out for the registration, and we hope you will join us then. I'm not seeing any additional questions coming through, but I'll give it another moment. Also, as a reminder, there will be poll questions at the end of the session, so if you can take a few moments to fill those out. We always appreciate any feedback that we receive as we continue to try to make this series as meaningful as possible. And I think with that, it doesn't look like there are any more questions. So I want to thank our presenter and everyone for attending. And please just take a moment to fill out the poll questions. And have a great day.